This procedure begins with removing the lenses from 2 to 4-day-old rats and cleaning them of any adhering tissue. The posterior side of the lens is identified and a small tear is made in the capsule. The capsule is then peeled downward and pressed into the bottom of the dish. This process is repeated around the entire lens. The lens fiber mass is removed by rocking it sideways. The peripheral epithelium is cut away, and the explants are then cultured. Finally, they are transferred to a test tube for biochemical analysis or onto a glass slide for microscopy.
Hi, I'm Peggy Zelenka in the Laboratory of Molecular and Developmental Biology in the National Eye Institute at NIH.
Hi, I'm Senthil Saravanamuthu, also from the Laboratory of Molecular and Developmental Biology in the National Eye Institute.
I'm Chen Gao, also from the same laboratory at NEI.
Today we'll show you a procedure for explanting and culturing lens epithelia from newborn rats. We'll show you how to remove the peripheral region of the epithelium, which contains cells in the early stages of differentiation. Then we'll show you how to induce the remaining central lens epithelial cells to differentiate to form lens fibers by exposing them to FGF.
We use this procedure in our laboratory to study the timing of events involved in the terminal differentiation. We harvest the explants at various times after adding FGF to the culture medium. Then we analyze protein expression by Western blotting, RNA expression by RT-PCR or microarrays, and immunostain the explants to determine the subcellular location of proteins of interest.
So let's get started by moving lenses from newborn rats.
To begin, use microdissecting scissors to remove the lenses from the eyes of euthanized 2 to 4-day-old rats by making a small posterior incision and pressing on the opposite side. Transfer the lenses into a 60 mm plastic tissue culture dish containing 5 ml of warm, sterile suspension medium. Using 0.1 mm tipped tweezers, clean any adhering tissue from the lenses and transfer them to a second 60 mm culture dish containing 5 ml of sterile suspension medium. This helps to reduce contamination. With the tweezers, transfer the desired number of lenses to a 35 mm culture dish containing 2 ml of sterile unsupplemented medium. As many as 12 explants can be made in the center of a dish this size. Any extra lenses can be stored in the second 60 mm dish from earlier and placed in a tissue culture incubator at 37 degrees Celsius up to 1 hour before they are used to make explants. Now identify the posterior side of the lens.
These two I can tell are sitting with their anterior side up. I'm going to turn it on edge so you can look down it and you'll see the cold cataract, which is an opaque area in the center. The side farthest from the opaque center is the posterior side. So I turn it over on the posterior side. On the posterior side, you can also see these striations, which are the remnants of this tunica vasculosa lentis. It's a blood vessel pattern around the periphery of the lens. That also marks the posterior side.
Correct identification is critical since this is where the capsule will be opened. Opening the lens on the anterior side will tear the epithelium. There are many ways to identify the posterior side. First, it is rounder than the anterior side, which is slightly flattened. Second, newborn rat lenses form cold cataracts as they cool to room temperature during the dissection. If the cold cataract has not yet completely filled the lens, the posterior side is the side farthest from the opaque region. If the opacity has filled the lens, warming the dish to 37 degrees Celsius for a few minutes will reverse it. The posterior side can be identified as the cataract reforms upon cooling. Third, vestiges of the tunica vasculosa lentis may be visible on the posterior side. Finally, the posterior suture may be visible on the posterior side. Once the posterior side has been identified, turn it upward and support the lens with the left tweezers. Then pinch the posterior capsule with the right tweezers to produce a small fold. While holding the posterior capsule with the right tweezers, grasp the capsule fold with the left tweezers and pull the two pairs of tweezers in opposite directions to make a small tear in the capsule. While grasping the edge of the retracting capsule with the tweezers, pull it downward, first on one side, then on the other, pressing it into the plastic of the dish with the tweezers. Repeat this several times, moving all around the equator of the lens until the capsule is firmly attached to the plate at many points. Holding the capsule in place with the left tweezers, gently rock the fiber mass with the right tweezers to break the attachment between the fiber cells and the epithelial cells at the lens equator. Now gently pull the fiber mass away, rolling it off the capsule, which remains attached to the bottom of the dish. Repeat this process with all the lenses in the dish. Fiber masses can be discarded or frozen and saved as a source of lens proteins for other studies. With microdissection of whole epithelial explants complete, let's look at microdissecting and culturing central explants. Using a sterile scalpel, trim away the peripheral epithelium, which contains cells in the early stages of differentiation, leaving a central square approximately 2 mm on each side that consists of central epithelial cells only. Transfer the dish containing the central explants to a biosafety cabinet. Wash the explants 3 times with sterile PBS and once with 1 milliliter of fresh equilibrated suspension medium. Next, add 2 milliliters of culture medium to induce differentiation, and place the explants in a humidified tissue culture incubator at 37 degrees Celsius, 5% carbon dioxide. The culture period may last as little as a few hours or as long as 2 to 3 weeks, depending on the parameter being studied. The medium should be changed every 2 to 3 days. With microdissection and culturing complete, let's begin harvesting the explants for analysis. At the end of the incubation period, remove the culture medium. Using microdissecting tweezers, gently loosen the edges of each explant. Grasp each explant with the tweezers and transfer it to a tube containing about 100 microliters of SDS lysis buffer for protein analysis or RNA later for RNA analysis. Agitate the tip of the tweezers in the solution to ensure that the explant does not stick to the tweezers. The explants are now ready for analysis of protein or RNA. Explants may also be analyzed by immunofluorescence. To begin, rinse explants briefly in PBS. Fix the tissues by adding 4% paraformaldehyde for 30 minutes at room temperature. Remove the fixative and replace it with PBS. Fixed explants become somewhat stiff, allowing them to be lifted and transferred to glass slides for immunostaining. Place a small drop of PBS, without calcium and magnesium, on the slide to help position the tissue and prevent curling. Using microdissecting tweezers, lift the explant and insert it into the drop on the slide. Without touching the explant, carefully use a paper wick to remove the liquid and flatten the explant on the glass. Let the tissue dry in air at room temperature for 3 to 5 minutes. The explants are now ready for immunofluorescence analysis. Shown here is an example of a microdissected whole epithelial explant that has been immunostained for N-cadherin, a marker for cells in the early stages of differentiation. Only the cells in the peripheral region of the epithelium are positively stained for this protein. Cutting away the peripheral epithelium as described in this protocol leaves a central explant which contains few, if any, cells that express N-cadherin. This is shown by Western blotting the central and peripheral epithelial tissues for N-cadherin. Western blotting for GAPDH was used to confirm that the protein samples were equally loaded. The timing of events associated with differentiation can be determined by culturing central explants for different periods of time. Central explants were cultured for 2, 5, 24, and 48 hours, in medium with or without 100 nanograms per milliliter FGF. The explants were harvested at the indicated times and transferred to SDS sample buffer for analysis by Western blotting. The results show that N-cadherin protein expression is induced after 24 to 48 hours in culture. Western blotting for GAPDH demonstrates that the protein samples were equally loaded. Immunostaining central explants cultured for 4 days in the absence or presence of 100 nanograms per milliliter FGF shows that N-cadherin immunofluorescence is strongly elevated in explants that were exposed to FGF. Immunofluorescence also shows the subcellular localization of N-cadherin at the cell-to-cell boundaries.
So we've just shown you how to prepare and culture lens epithelial explants for studying differentiation. The main advantage of this system is that the cells differentiate synchronously. This makes it possible to determine the sequence of events. In addition, we can add specific growth factors or inhibitors to the medium to activate or block particular signaling pathways.
When doing this procedure, it's important to remember to guard against contamination by dissecting in sterile medium in a clean environment and by washing the explants thoroughly before culturing them.
So that's it. Thank you for watching. Good luck with your experiment. Bye.
Bye.
Bye.